talks. So first up, we have Leah Fridley, and she is doing her PhD in environmental science at the University of Lausanne. Do we have Leah? Very good. Welcome, Leah. So Leah applies advanced statistical methods in geophysics and enjoys the challenge of making sure her statistical formulae are rigorous, but also feasible to use practically. As a physicist, I could learn a few lessons there, I think. It's a very sensible approach. In life, at least in the UK, begin with the weather. I thought Leah would be the perfect person to kick us off with her talk entitled Rain Before 7, Fine Before 11, Statistics Edition. Over to you, Leah.
The more general structure, which can be applied to whole Switzerland and not only to local places. This structure could, for example, include a fact like precipitation forecasts for mountain places are always too low in January. And a whole collection of such facts makes it then possible to post-process weather forecasts in any place in Switzerland at any time of the year. And even though this approach did not reduce the number of raining days, it did reduce the times of me being drenched. And for the times it is not working, as Vivian Green, a British writer and expert for roll houses, said, life isn't about waiting for the storm to pass. It's about learning how to dance in the rain.
Thank you very much, Leah. Excellent. Stay with us for now, Leah. We have a bit of interaction with the judges panel.
Sure.
But just to say, it's proof that statisticians have friends. There you go.
I was actually thinking about including this joke, and then I decided
Wow.
again.
Thank you for saving it for me. So maybe we can start off with Tina then.
Sure.
Yeah, I was wondering, so do your friends regularly call or family, and do you then explain it to them how today, and what do they say?
You mean when they tell me how
Yeah,
the weather was, or
when-
like in general?
When they want to plan their trip.
Yeah, that's really, I mean, I have like some kind of doodle when they can say when they plan their next trip. And if they really want to have a good weather forecast, they call me, yeah.
Nick?
I have a bit more of a content question for you.
Yeah, sure.
When you develop your models, you said you use probabilistic models,
Yes.
right? Do you develop those by data only from your friends, or do you go to any other sources for data? Because that sounds a little bit skewed.
There are also some weather stations in Switzerland, and we can use them.
Okay, I would definitely. Thank you.
I have a question. Where do you find your information? Which is the most accurate source for you? If you looking, would you say the Apple phone, or when you're looking for weather forecast yourself, or do you make them yourselves?
That's a really tricky question. So, like in this project I was working together with Meteor Schweiz, like the federal one, and so I'm kind of really connected to them, and I still check their forecasts.
Very good.
Excellent. Thank you very much, judges. And Leah, another round of applause. Well done. Thank you. Now, next up, we have Anna Wieber, who is a PhD student in biophysics at ETH Zurich. Welcome, Anna. Yeah, why not? So, Anna, you work with mathematical and computational methods in immunology, and she combines her passions for molecular medicine and physics, which sounds very, very nice. But what's the other thing that people like to talk about after the weather these days? I was wondering, we've started with the weather. The next thing people talk about is the pandemic. So we're very lucky to have Anna here to talk to us about viral immune evasion, a game of cat and mouse. Over to you, Anna.
Do you still remember the classic Tom and Jerry cartoons? The little mouse Jerry is constantly being chased around by the big cat Tom. But in the end, Jerry always manages to outsmart Tom and persist in a safe hiding place to continue the endless fight in the next episode. Amazingly, there is a very similar game of cat and mouse playing out within ourselves. At least every second one of us has a little Jerry hiding in their bodies at this very moment, and it's called Cytomegalovirus. Despite its megalomaniac name, this virus is hardly a supervillain. In all likelihood, you never felt any symptoms at all from the infection. But cytomegalovirus, it's more like a sneaky ninja. Like Jerry hides in his hole, cytomegalovirus hides in our cells. But it's chased around by our immune system. All cells regularly have to show their contents to a special immune police force. And if they find virus inside, they will immediately shut down the infected cell. But cytomegalovirus is a master of deception and disguise. When it infects a cell, it does everything it can to prevent this cell from showing any signs of its contents to the police. That way, the virus inside remains invisible. Well, the immune system had a long time to learn about that clever trick. So to counter it, we have the natural killer cells, which, well, kill all cells that refuse their contents. Yep, the immune system does not take disobedience lightly. And so the battle goes back and forth. To avoid the natural killer cells, cytomegalovirus makes the cells show fake contents to the police. And when immune cells still get suspicious and send out calls for reinforcement, cytomegalovirus catches and destroys the messages. And it can even send out fake ones, signaling that all is well. With all these tricks up its sleeve, cytomegalovirus evades the immune system so well that once we catch it, it stays with us for the rest of our lives. Over the millennia, this endless battle has forced our immune system to become better and better. And what's more, some researchers speculate that contracting cytomegalovirus in early childhood may actually make a baby's immune system stronger against bacteria and could even prevent allergies. So in the end, like with Tom and Jerry, you begin to wonder whether the perfect enemies might actually have become best friends.
Fantastic, Anna.
Thank you.
I went through the motions there. I sort of hated the cytomegalovirus, and then at the very end, actually, it's not so bad.
Maybe it's not.
Maybe it's not. Nick, over to you.
Right. This might come a bit strange. So what is the project that you are actually doing with cytomegalovirus?
I'm not personally working with
Okay.
cytomegalovirus. I'm more working generally with the immune system and trying to develop models for this immune police, actually. That's the T cells. So that's actually where my personal research is focused on.
Okay, so it's antigen presentation and
Yes.
ABC. Okay, right. Okay, thank you.
Immune police. I like it.
Yeah.
It should be a US TV show, I think. Joe?
What does cytomegalovirus actually do? What is its purpose in life?
Oh, its purpose in life. I mean, its purpose in life is to infect as many of us as possible and to spread from one to the other. And it does that very, very well. This is why at least every second one of us has it. But yeah, it doesn't, as I said, it usually doesn't do much to us. It doesn't have bad symptoms, except when your immune system isn't so fit and can't control it so well. So, for example, newborns, when they get it basically already in the womb from their mother, that can be a problem, or sometimes old people when their immune system gets weak, then they get trouble with this virus. But most of the time, I mean, yeah, half the room has it, and it's not doing anything that we notice.
Thank you.
And Tina?
So, yes, what is the biggest challenge when communicating research to a lay audience?
The biggest challenge, I feel like the biggest challenge is to reach people who aren't already interested in science in the first place, and to still engage them and make them interested, and yeah, make them listen to science talks, for example, in the first place. Because usually the audience at something like FameLab is people who are already interested, right? And I think the harder part is reaching those people who wouldn't naturally attend such events.
That's why we need to make the Immune Police a TV program.
Yeah, that would be that would probably be a good idea.
I'll give you 50% commission for that idea. Thank you
Awesome,
very
awesome.
much, Anna.
Thank you.
Okay, so our next finalist is Joanna Ashok, and she is a master's student in chemical engineering at EPFL. Come on up. Welcome, Joanna. So as you might tell from her outfit, Joanna loves food, not just for the joy of eating it or the artistry of cooking it. I mean the apron. Not just for the joy of eating it or the artistry of cooking it, but also because of all the science going on behind the scenes when you make food. So today, Joanna will talk to us about chilies, pain or pleasure. Now that's a hot topic indeed. Over to you, Joanna.
A while ago, I got a really angry text message from my friend. You see, I'd left her some food, but she ate it and ended up with a stomachache, fever, and diarrhea. I was really apologetic, but also so confused. I promised her I was going to use as little chili as possible. Turns out, I still ended up causing her pain on the way in and the way out with that food. But I love that. But why? Why did we have such different responses? Turns out, the culprit is a little molecule. It's called capsaicin. You might have heard of it. It's present in chilies, and it's an oily substance. Capsaicin has an interesting structure, and this binds to special pain and heat sensors, pain and heat receptors on our tongues. This is just like a key fits into a lock. This then causes neurotransmitters to flow between neurons and send a signal all the way up to the brain. Oh no, we've eaten something way too hot. It's so painful. And so our brain then causes us to feel that burning sensation of pain on the tongue. Unfortunately, it doesn't just stop there. If you've eaten chili, you know that it hurts you twice. And this is interesting because you see, capsaicin is not digested or transformed by the body, but instead it just goes through our digestive tract. Our intestines and our anus have similar pain receptor cells to our tongue. So that means capsaicin has a chance to burn us once on the tongue, but then again on the gut and the butt. So that is quite painful for most of us. Yet, so many of us love chili. Why? Do we just not have pain receptor cells? Well, it's also because when our brain is experiencing the stress of the capsaicin on our tongues, it's also releasing endorphins. Endorphins are feel-good chemicals, also released during exercise and sex. And this can give people a natural high right after eating capsaicin. And people can start seeking that. It cements this relationship between pain and pleasure and makes you seek more capsaicin-rich foods. But if you're not like me, and you hate chilies, and you've just had something with a lot of chili in it, what do? Well, rule number 1, do not drink water. We've just talked about how capsaicin is an oily substance, so water and oil don't mix. Instead, try dairy, something with milk protein called casein in it. This then binds to the capsaicin, dissolves it, and washes it away when we swallow. It takes away the pain from the tongue. And if that doesn't work, honestly, just try to focus on the endorphins that your brain is sending out. Because at the end of the day, yeah, chilies, they do cause pain, but they also do cause pleasure.
Thank you, Joanna.
Would you like a chili?
No.
Okay.
I knew you were going to ask me to eat chili. I am the world's least adventurous hot foods eater.
You should just have some milk.
It'll
You'll be fine.
Capsaicin overdose. What if you're lactose free on a diet? Is there anything else you can use?
You would use something that is still, capsaicin is an organic molecule, and so you don't want to use anything like water, which is a polar substance, but you want to use something that dissolves it. So instead of that, you can try other things, but try something not too fatty. That's what you have to stay away from, because the fat will just make it move around in your mouth, and that can be problematic. So that's why when you have those wings with all that grease and that butter, delicious, but painful.
Words of the wise, I think. We also have some
there's a
chili on the floor.
Yeah. My bad.
I'm probably allergic to it even from here.
I've got plenty more. Where did this came
It seems so. Joe?
At the beginning, you said that you had a friend who hates chilies, you love chilies. Do you know the biology, or can you have any explanation for why one person likes them and one person doesn't?
Yeah, so some people, repeated exposure to capsaicin, actually, it does cause pain in our tongue, but the thing is, over time, quite quickly, we get desensitized to it. And that's actually really interesting. It's a fact that I couldn't put in my talk, but now I can say it. It's the idea that capsaicin can also be used as a pain relief treatment. So while initially it's that strong, all the neurotransmitters are being sent to the brain, sometimes you can just put capsaicin over joints that have arthritis, for example, and then you just cause the neurotransmitters to just exhaust themselves. So all of it's just been spent. It's that short amount of pain, but over time it provides pain relief, because now the neurons have nothing more to send, saying to the brain, oh my goodness, we're in pain. Yeah.
Thank you.
Nick?
Yeah, so I'm half Caribbean, so this really resonates with me, this talk. And I've got a question about that. Why do you think is it that close to the south or on the equator seem to have, other than the fact that chilies and peppers, Scoville peppers and Scotch bonnet peppers and all grow around there, but other than that, why do they have such an affinity for something that actually makes them hot?
So that is actually why. So we experience this heat, and we experience that on our tongues. And so as a result, we quite often start to tear up, the mucus glands start to secrete mucus, and we start to sweat. That's the key there. Because in hot cultures, a lot of times when you're eating chili, you're causing yourself to sweat, and so you're able to remove the heat from inside your body. It's the same reason why they also often drink hot drinks as well. You think you only want ice water there, but no, sometimes you do want a tea and a hot coffee in the dead of summer. So,
Okay.
Yeah.
I'll remember that for the next family gathering.
Yeah.
Top tip. Tina?
So food is a research topic for you as well, and do you have special reactions from family and friends when you are talking about food and research?
Yeah, well, so my research has actually been on processes more than food. Food is more of my hobby in the kitchen, and I'm really interested in being able to substitute ingredients just based on their chemistry. So a lot of times they'll patiently listen to me talk for like 15 minutes about food, and then they get to eat. So there, it's okay, you know. It's the cost, it's the price they have to pay. So usually they're just happy to eat my food afterwards.
You bribe them with food to get you so you can talk to science.
Yeah, how do you think I practice for FameLab?
Yeah, no, it's a great strategy. It seems to work. Thank you very much again,
Thank you so
Joanna.
much.
Okay, so next we have Harini Lakshmi Narayanan, and she is a PhD student at the University of Zurich. So Harini studies biomarkers for monitoring disease progression in kidney cancer patients, and she's traveled the world for science, from India to Switzerland via Boston and Heidelberg, but we have become unstuck by a table,
Yes.
regardless of all the travel. There we go. Okay, so Harini's talk is called Looking at Cannibalism Under a Microscope. Now, even though I just finished 3 seasons of Hannibal, the TV show, the other day, I have no idea what to expect here, but I'm very interested to find out. So please take it away, Harini.
All right. Death. It is not an easy conversation to have, and yet researchers around the world wonder about this every day. No, not the big philosophical questions surrounding death, but the very specific details at the molecular level. You see, cells die in our body every minute and in remarkably different ways. Entosis is one such type of cell death that involves cannibalism. Yes, you heard that right. Welcome to my talk. This is officially a Hannibal lecture. Entosis is a type of cell death where one very healthy cell, specifically an epithelial cell, engulfs its neighbor to form cell-in-cell structures. So let's imagine that there are 2 cells. Happy, healthy, friendly. Like neighbors do, they decide to play tug of war. Why not? So there's a lot of pulling and pushing. Both of the cells are competitive, and the game is not ending. So now they're tired and fatigued and glucose-starved. This cell decides it's had enough, releases tension, colliding with this cell, and now they're stacked up like Russian dolls. But wait, this is not normal. Why did this happen? Well, the biophysical forces acting on this unit was unequally distributed, right? So now they're stacked up one inside the other. But what happens now? It's not normal for this to happen either. Sometimes the inner cell can go on to remain viable and even undergo cell division, but most of the times, the host cell ends up eating the inner cell. Viewer discretion is advised. This is gory cannibalism. So like you and I need carbohydrates and proteins to grow and survive, the host cell, which just is tired, and so kills the inner cell and uses the glucose and the amino acids from this cell to continue growing. Okay, let's take a beat. Now you know that humans are cannibals after all, thankfully at the cellular level. But why is this important? Entotic cells are seen quite frequently in late-stage and aggressive cancers and are speculated to play a role in the progression of the cancer. Simplistically, this can happen 2 ways. Either a tumor cell engulfs a healthy cell, causing tumor promotion, or a healthy cell engulfs a tumor cell, suppressing the tumor growth. In reality, it is not just this binary combination. There are several more factors that play a role in deciding the fate of the cancer tumor. So while I talk to you today about entosis, to truly understand this type of cell death and the role that it plays in cancer progression, and also how we can use it to treat cancer, scientists will have to continue to look at entosis and cannibalism under a microscope. But in the meantime, the entotic food stall in our body will continue to serve cannibals, so plates up. Thank you.
Thank you, Harini. Brilliant. Excellent stuff. Hannibal lecture, that will be pun of the day, I think.
know, I was
love
so
it.
proud of it.
Brilliant. I didn't come up with it myself, so fantastic. Okay, so Nick, perhaps you want to go first?
Oh, so many questions.
Okay.
No, just one question. So, do you envision some kind of exploitation of entosis for the treatment of certain tumors?
Yes, actually. So entosis, simplistically, I mentioned it as tug of war, but this is something that is also driven by mitosis, which is a type of cell division, right? And cancers divide a lot. So a lot of drugs which block mitosis can also help either coax one cell into the other, because in mitosis, the cells which are adherent to the tissue through the extracellular matrix, pardon the jargons, they are either fluid or rigid, depending on the case. And so when you use these drugs that kind of target mitosis for cancer, you can actually coax, or it's rather speculated that you can coax one cell into the other. It is a double-edged sword, because you want the right cell to be coaxed into the other, but scientists are doing a good job, I guess, researching about this, so yeah.
Okay. And this is kind of a new field now because?
Yes,
okay.
it's relatively new in terms of exploring the
Yeah.
field, but it's been observed
Yeah,
by
yeah,
pathologists for a really long time now, actually.
Okay. All right. Thank you.
Tina.
Yes, you participate in FameLab, and what would you like to pass on to other scientists?
That they should read about their science. So I don't work with entosis particularly, I work with cancer, but I'm still wanting to talk about entosis. And I really think that we should be having more conversations about science. It's geeky, it's nerdy, but it's also a lot of fun, and maybe you can throw a pun once in a while and then it's good to go.
You definitely won that battle, at least. And Joe?
You talked about the way that the healthy cell can eat or go inside, and vice versa. Do we know which way, or what will actually trigger which one wins, if you like?
I don't think that's so very well understood yet, but that's something that scientists are still working over. There's literature, but far and few, so that's still something that needs
Something to look forward to.
Yeah. So I think when scientists see that there is one cell type of maybe we should study this. And so they're studying
So do you
entosis.
think this could become a biomarker for, as a research biomarker, looking for these cells that are inside other cells?
So I'm not sure if that could be a case because although it's frequently seen, the frequency of these cells in like the larger population is still not as high as it.
Bake a muffin with some blue food coloring. You eat it, and you note down the time. And then you wait until your poo turns out to be blue. And then you note your time again. This is the transit time food needs to pass through digestive tract. We see that people which have very fast digestions below 15 hours, or people that have very long digestions over 50 hours, have certain diseases and are more risk of diseases. Like, for example, people with cardiometabolic health problems have elevated levels of a certain bacterium. They also have a very long transit time, which is very promising, this correlation between the two of them. Because next time before you run to the doctor to check on your health, maybe bake yourself a muffin.
Thank you, Dubravka. Brilliant. Have you brought muffins for all of us? We
No,
could do an
actually-
experiment. Good statistics in this room.
I did it with all my flatmates, with all my family, and they really liked the experiment.
I'm sure they did. Sign me up. Sounds
only-
good. Tina.
Just pass on to Nick.
Yeah, sure.
Pass on. Okay, that's nice. That's a good,
Yeah.
that's a good
Yeah,
word to use.
sorry.
A good metaphor.
So, I'm trying to understand here. So, the muffin is supposed to take to give information upon elimination, to give information about the dominant cultures or the dominant bacteria inside your microbiome. So that's the idea, or to tell you if there's a mutation, or what
They
exactly
tell you
you're
if
looking
there's
for.
a balance
Okay.
or an imbalance. Because this time that food needs,
Okay.
if it is too slow or if it is too fast, gives indicators that there's something going on which is not good.
Yeah.
And after this test, you do a DNA sequencing.
Okay.
So it's like the pre-test, you do a pregnancy test before you go to the doctor. And this is very interesting because people that cannot afford it and don't know what is going on with them, they can just test themselves at home.
Okay. All right. Thank you.
Joe?
I love the idea that there's sort of a simple health measure that most of us don't know about. How do you sort of approach sort of science communication? How would you ultimately like to make sure that everybody, what was, literally getting people to check their transit times?
I think you need something that people are used to, that is something that they do every day. Like, for example, we all poop, I guess, and we all eat. And something that people can adapt to and know how to use at home, because science sometimes, I think, is very complex. And the complex things are not the ones that, at the end, are put into practice. It's like the complex makes us understand how to put something in practice. So make it for the people something that, in words, especially in words, that they know, and with some humor.
Brilliant, thank you.
Thanks, I agree. Should we come back to you,
yeah,
Tina?
sure.
Cool.
So, how do you convince your friends and families, or did you convince your family and friends to eat this cake, this blue cake? Or
I don't know, would you?
yeah, sure.
You see? I mean, all of them, I told the story, and all of them were like, so, and when can I eat that? So I baked a bunch, and they all wanted to try it, because knowing something about what you eat, and if you're healthy, this is something which is rather new, and also microbiome and what's going on there. So people are very curious, and yeah, everybody likes to play with food. So they're like, these were free voluntaries.
Nothing to do with the muffin bribe at all, you think?
No.
Okay, thanks again, Dubravka. Fantastic. All right. So our next finalist is Prina Yadav, and she's a PhD student at the University of Zurich. You're also using the table. I was looking for you. Do you need a hand? Thank you. Yeah. Very good. Okay. So Prina's a chemist and a health enthusiast. And when she's not in the lab, you can find her doing body combat or dancing. So what I didn't know is if you do dancing and combat at the same time or not. But either way, I'm very impressed. So today, Prina will talk to us about resistance versus tolerance. Over to you.
Thank you. Let's talk about why I can't see your faces today. One word, coronavirus. But this scenario, two questions are very critical to ask. First, how are we going to prepare our bodies to fight a future pandemic if it's awaiting for us? Second, is there a possibility that we can avoid a pandemic in future altogether? The answers lie here. Bats. Yes. Also, the reason that I'm dressed like a woman in black today, but bats are the source of coronavirus, that are considered as the source of coronavirus. But in fact, in presence of a large amount of virus, bats don't get sick. However, from our own experience, we know that humans, when we expose them towards this virus, we tend to have symptoms. We might even get to hospital. But let us dig deeper into that. Consider a human cell. Now, in presence of the virus, the cell senses a danger. And what it does, it activates large amount of proteins, molecules, which are molecules called as NLPR3, which in turn then activates another set of molecules called IFNs. These IFNs stimulate an inflammation response in your bodies, by which you can actually fight the virus and you get sick, you get swollen, or other type of sickness. But if this regulation is not, if this type of response is not regulated properly in your body, you might have different kind of diseases, even tissue damage, and worst case, cancer or even death. On the other hand, in bats, this activation of this NLPR3 molecule is less in presence of virus, thus limiting its inflammatory response caused by these IFNs. As a result, there is no tissue damage, no sign of diseases in bats. Instead of finding a way to fight this virus like we are trying, they just tolerate it somehow. Viruses don't know any bounds when it comes to evolution, and so do bats, thus making them very good for studying to prevent future pandemic and help in drug development as well. I, as a chemist, believe that it requires a collaborative scientific approach so that we can see whether to resist or tolerate to achieve global One Health system. What do you think? Thank you. Thank you.
And a really nice use of props, I thought. I love the massive cells with little spinny bits. Very nice.
Thanks.
Who should we start with? Joe?
You were talking in the very relevant case study of COVID. I know that bats, according to you, do not get COVID. Do they not get any? Do they not suffer sort of the ill effects of any viruses that they carry?
There are exceptions, like everywhere, but most of the viruses that they inhabit, they actually just like coexist with them. They are reservoir of a lot of different kind of viruses.
Nick?
So, I'm a bit unclear on your project. It's on the comparison between the human cell and the bat cells. Are you, as a chemist, are you trying to use the signaling pathway from bats and mimic that into human cells? What are you trying to do there? Because but this is my fault. I might have just missed
no,
that.
I mean, I'm a chemist, but that's what, as Harini mentioned, I've just like as a scientist, I'm interested in health enthusiasts that Donald also mentioned. So this is one of the topics that interests me at the moment, and there has been a lot of research done at MIT and various other institutes for kind of, there are difficulties at the moment because bats, for reagents and stuff to study bats. Yeah. So it's very limited study at the moment, but it's very promising.
Okay.
for like we can have drug targets from this kind of like studies. We even know how they are resisting this stuff or tolerating this stuff, as I said. So we can learn something so that the spillover of the viruses won't happen from bats to us.
Okay, so the point is to prevent zoonotic viruses from transferring to. Okay.
Yeah.
All right.
Yeah.
Okay. Sorry, just I got distracted with your little your purple and yellow ball there.
Tina?
Yeah, you said we should go towards global health system, and now you're at the University of Zurich, and I'm sure you're collaborating also with others. So how do you manage to collaborate, and why?
As I said also before, like I'm a chemist, so we already collaborated on like 2-3 different projects. So one was where we developed a sensor for some anions, and then we collaborated with a biologist who is in Paris so that they can do the cell studies for us, so that if it works or not. So yeah, we try to develop a meaningful relationship to see what fit we can do with which kind of group.
Thank you.
Excellent. Thanks again, Prina.
Thank you.
Congratulations. Give you a hand. These are super cool.
Thank you.
Okay, so coming up next, we have Hugo Cui. And Hugo is a PhD student at EPFL. Now, Hugo studies how the tools of physics can be used to understand machine learning algorithms. Now, as a physicist myself who uses machine learning algorithms, this sounds interesting, because usually we get the machine learning to help us do physics, not the other way around. So I found that was quite interesting. Now, today, Hugo's talk is called It Breaks in Threes. So please, take it away, Hugo.
Yes.
You also need the table.
Let me tell you a true story. It's the story of Lázaro Borges. Borges is one of the best pole vaulters in Cuba. He made it as far as to qualify for the London 2012 Olympic Games. On the day of the semi-finals, Borges takes a deep breath. He begins to run, accelerates, plants his pole to the ground, prepares to jump, when suddenly his pole breaks. And not just cleanly into 2 halves, mind you, it literally snaps into 3 pieces. That was pretty dramatic to see, but it's explainable by physics. Physicists have indeed shown that any rod-like structure, such as a pole, can never just break into only 2 pieces. It always breaks into at least 3. And they discovered that not by stoning a pole, but something much shorter than a pole, much thinner than a pole, but it has the same physics as a pole. Namely, a spaghetti. So today I'll be telling you about the physics of breaking a spaghetti. Please do try to reproduce that at home. Take a spaghetti, bend it, break it, and count how many pieces you get. Sorry for that. You always get more than 3. Why is that? Sounds like magic, right? Why is it impossible to just break a spaghetti simply in 2? Well, let me explain. When you bend the spaghetti, you increase its curvature until it exceeds the maximal curvature the spaghetti can bear, and the spaghetti breaks for first time into 1, 2 pieces. But that's very far from being the end of the story. There is now a free extremity that is held by neither of my hands, and that is therefore free to oscillate, thereby creating waves that propagate along the spaghetti. But my hand is still there, so these waves cannot propagate any further and get reflected back the other way. There are now 2 waves that propagate along my spaghetti, the original wave and the reflected wave. And they add up, creating even more distortion in my spaghetti. This phenomenon is known as the interference of 2 waves in physics. And because of interference, the distortion shoots up again at some point of the spaghetti, causing it to break for a second time. And I now have a total of 1, 2, 3 pieces. That's why I get at least 3 pieces, because of interference. And remember, this is true for any rod-like object, not just a spaghetti. So whether you are an engineer trying to craft some carbon nanotube fibers, or maybe a biologist interested in the mechanics of microtubules in human cells, or just an industrial trying to make an unbreakable pole for athletes like Borges, remember, start by breaking a spaghetti. Thank you.
Excellent.
Thank you.
I love how much spaghetti you brought up here. It's like we can all give it a try afterwards, I think.
I mean, if you want to try,
Yeah. Also,
please feel free.
enough for dinner left over afterwards. Fantastic. Nick?
Yeah, you can put it with the capsaicins or the peppers afterwards and make a meal, right?
Yeah, sure.
So, how can you avoid it?
How can I avoid?
How can you avoid a rod?
There's been very recent research in MIT, I think, that discovered a very, very, very subtle way to avoid it. And they tried it with lots of spaghetti barrillas, number 7, 1, 5. This is 5, by the way. And they discovered that by applying a torsion to the spaghetti prior to bending it, you can avoid it. So I invite you to check the papers. But it's an active research domain in pasta physics.
Wow.
The MIT's on it, so
That's awesome. Yeah, thanks.
I think so.
Twist and bend. That's a
You have to
good
twist
life philosophy. Joe?
Tell me a little bit more about the applications. I know you said there's lots of different applications, but can you maybe just give one example of maybe in the biology setting you talked about?
Or maybe, yeah, or the nanofibers,
Yeah, and the
for example.
nanofibers,
For example,
great.
you can do nanofibers with silica particles, and you can add some polystyrene particles and then burn that so that you get some voids. And that affects the mechanics of your carbon fiber, right? And you want to understand how much the percentage of void in your fiber controls how it breaks, how brittle it is, how elastic it is. And so that's when you can apply that theory to understand and predict how is the mechanics of your carbon fiber with the spaghetti.
Thank you.
Tina?
Okay, as you have heard, I work also with citizen science, and this is really to include citizens into science,
Sure.
either to collaborate or co-create. And I have questions. Do you see any possibilities to collaborate or co-create a project with citizens in your domain?
This is not my domain. I don't
Yeah.
work in pasta physics.
Yeah.
Sorry. Do you mean like creating an event and having people interact and try by themselves?
No, really, in your research,
in
to
my-
do research with citizens.
No, I didn't. I'm doing research in artificial intelligence, which is a bit far away, so no, I'm not working with citizens.
So it's too complicated to include citizens in your research.
I wouldn't say so. We can do some coding. I haven't done it so far, but it might be possible to get some, for example, sessions for introduction to coding for kids that they can play and see by themselves. Of course, I think this kind of initiative has already been done, but sure. I haven't been doing it. That's what my
Okay.
answer to your
Yes,
question.
thanks.
Trick is to cook them pasta for lunch first and then get them cooked afterwards. So, yeah, thanks again, Hugo.
Thanks.
We can clean it up later. Okay, so next up we have Caroline Sommerts-Rambo with an assortment of little friends. And she's a postdoc at the University of Basel. So Caroline studies animal behavior and I believe specializes in fish. So she's currently studying cichlid fish in Lake Tanganyika in East Africa. And I actually looked it up. There are over 250 types of cichlid in that lake. So that's definitely enough to keep anyone busy, I would say. So Caroline's talk today is entitled, Are You of a Curious Nature? Personality from an Evolutionary Perspective. Over to you, Caroline.
Imagine the face of my 90-year-old granny when I told her that I earn my money with studying fish personality. She was like, oh dear, we love you anyways. Now, 3 years later, unfortunately, I can't share the results of my project with her anymore. But I can share with you. So please try to picture the following scenario. You're standing with your feet in crystal clear lake water, and a group of tiny fish is coming towards you. Some of them don't really dare, you can't even see them, but some are coming closer, and 1 or 2 even nibbling on your feet. It's the same situation for all these fish. It's some human legs in front of them. Still, they all react differently. And this is because individuals in general differ in their perception of risk and also in how curious they are. This is what we call animal personality, and it's related to the concept of human personality. Now, in our research project, we looked at the genes underlying these behavioral differences. And we found one gene that seems to be important in explaining curiosity differences in our fish. Let's give this a little bit of an evolutionary perspective. All creatures on this planet are related to each other. It's just a matter of how closely. Now, if we compare two different species with one another, then the probability of finding similarities between them is much higher when they're closer together on the tree of life than when they're very distant. Sounds logical. In our case, if the fish on the tree of life are about here, then in terms of distance and relatedness, the mouse, an important study organism in science, would be quite far, like here, and the human is here. Don't be shy. Close, not so close. Remember, we might have found a curiosity gene in fish, and after intense studying of the scientific literature, we assume that the same gene also affects curiosity in mice. But look at them, they're quite distant, so this is a rather surprising finding that these distantly related species share this cool similarity. And this is so exciting, because now it's only such a small step towards the human. And wouldn't it be cool if there was a global personality gene? You know, this whole personality research also always makes me think about animals as individuals. It's not just the fish. It's this particular fish that's curious enough to nibble on my foot, or that shy one over there. And I think this perception affects a lot in how we treat animals in general. So next time you're in the lake and the fish is coming closer and you are coming closer and you're both curious about each other, you might remember that you have much more in common than meets the eye. Thank you.
Thank you very much, Caroline. Fascinating and extremely cute little creatures, I think. Okay, so shall we start with Tina?
Okay. What are your lessons learned now from preparing for FameLab?
Well, my lesson learned for preparing FameLab is just concentrating on the message. I really want to communicate this. But me personally, I am maybe not the personality to be on a stage, so I just needed to practice a lot. And I was doing this in the aquarium, so I talk to my fish until they know by heart. Now, this is my personal lesson.
They could give the talk eventually.
They could now, trust me.
And Joe?
So interesting, you talked about the fish and the curiosity gene, the idea of that being a personality trait. Did you look at other personality traits in the fish?
Well, there's the big 5 personality traits. Apparently, you know a little bit about this. So it's risk-taking, it's curiosity, aggressiveness, activity, and sociability. In my project, I compared many species, not only individuals, but many fish species. So I needed to choose one of them that I would really find in all of the fish species. Not all of them are aggressive, not all of them are social, you know, and so the easiest one to pick was really curiosity. So I was looking more at this really specifically. Yeah.
Thank you.
Nick?
Which gene? Which gene is it?
It's top secret.
Okay.
The study is not yet published. I would really like to share all of the details with you, but I just can't.
Okay,
so
let me modify the question then. Definitely. Which is it? Is it a gene that affects cognition ultimately through? Because obviously, if it's a gene, then it produces. This has to do with a protein and so forth. Are there epigenetics involved?
Yeah. So it's
What I'm asking is curiosity correlated only to one particular gene, the presence or the activity of just one gene, or is the gene that drives a gene network, or?
Well, okay. So for sure, one message that I found with the analysis is that many genes are involved in personality. It's not only one gene that drives it, but there was one gene with like the perfect correlation to our behavioral data. But now we need to find out how big the impact of this gene is still. So I can't actually tell you. It's expressed in the brain. It has nothing, I don't think it has anything to do with cognition. Also, I don't think that personality and cognition are correlated. I did an own study on this showing that's not, at least not in fish. But it's related to neurotransmitters and brain and stuff like that. Yeah.
That's very fascinating. Thank you.
Keep our eyes peeled for the study then. Thanks again, Caroline. Do you need a hand or it's okay? Okay, our next finalist then is Elena Konova, and she's a PhD student at the Lausanne University Hospital, or SUV. So Elena studies neuropathic pain mechanisms, and before she moved to Switzerland, she studied in Manchester, London, and even Lund in Sweden. So today she's going to talk to us about adding insult to nerve injury. Ouch. Over to you, Elena.
I have to admit, I'm a clumsy person. I was trying to cook and I cut my pinky finger. The injury to my nerve is sensed by nociceptive nerves that send electrical signals called action potential. They come from my fingertip all the way to the spinal cord and then relay it to my brain to interpret, ouch, not again. I should really stop trusting myself with a knife. The pain will pass and the injury will heal. It wasn't the worst thing that could have happened. But had I struck a nerve, the nerve injury would lead to neuropathic pain. You'd think a cut nerve can no longer send signals because the pathway to the brain is severed. Indeed, you can no longer sense what your finger's feeling, but the injured nerve gained this annoying ability to send spontaneous signals whenever they like. In an attempt to repair themselves, the injured nerves alter the mechanism that tightly regulates when and how the action potentials are triggered. Other things like the formation of neuromas, scar tissue, and persistent inflammation can activate nociceptors to make spontaneous pain. Alongside these really annoying, attention-seeking, damaged nerves are neighboring nerves that were lucky enough to escape the knife. And these unsuspected nociceptors are there to add insult to injury. They innervate the other fingers in the hand and run along the arm, but at the dorsal root ganglion, the injured nerves and the non-injured ones mix, and this is where the signal of the nerve injury echoes to the other nerves. This is called peripheral sensitization. It makes the non-injured nerves hyperactive. They think they're helping, they're working harder, better, faster, stronger, but what is actually happening is that those intact nociceptors are overreacting to the weakest stimulation by sending many more signals at a high rate. This surge of action potentials to the spinal cord increases the activity of the rest of the pain pathway and eventually leads to chronic pain. So, effectively, the pain pathway has turned against you and you wish to turn it all off. Given these risks, I'm going to accept that I'm a clumsy person and this could lead to a disaster, and I'm going to ask someone else to cook for me.
Brilliant, Elena. Thank you. Thank you. Excellent. Harder, better, faster, stronger, while glowing like a Daft Punk music video. So I like that very much. Excellent. Okay, shall we start with Nick?
Yeah. Fascinating. I worked in neuropathic pain one long time ago. Oh, I'm sorry. Right.
No, you didn't.
That was the time. No, you didn't. You're lying. Yeah. It was a fascinating project. But what I want to ask was, how, does this relate to neuropathic? Well, ghost pain, basically. You know, the loss of a limb, and so that's.
To some extent, yes. So phantom limb pain. Most people might know about phantom limb sensations, where you feel like you have your arm still there when it's been amputated, but not so many people know that you can actually also feel pain. And these are caused by similar processes. The thing is that the rest of the pathway is still connected, and that's why you perceive the pain being in the limb that's missing.
Yeah. Okay. All right. Thank you.
Joe?
I love the way that you explained the way that you get this sort of, additional pain or sympathetic pain. In understanding it, is there a way of breaking that cycle? So is there something that we can use with this knowledge to prevent it happening?
So I believe that fundamental neuroscience is important to find new targets that can later on lead to new treatments. The difficulty is how that we believe that this is the first mechanism that initiates later on chronic consequences. And reversing those consequences are quite difficult. So maybe in having a preventative treatment before an operative treatment, an operation, this is how this sort of research could be applied in this context, rather than reversing chronic pain that's been already there for many, many years.
Okay, thank you.
Tina.
So when you are talking to a lay audience about your research, how do you know that they are really interested in it?
So it's a difficult question. Obviously, you try to read their expression and really know that it's a two-way communication. You want to be sure that the person is listening to you and trying to grab their attention in whatever way possible, like with blinking lights. It's really helping. But yeah, ultimately, you try to relate to them and to their personal experience, perhaps.
Thank you.
I agree. Thank you very much, Elena, again. All right. So believe it or not, we have reached number 10. Our last but not least finalist is Karen Poehler. Now, Karen is a postdoc at Agroscope, where she analyzes the impact of cattle, sheep, and goats on the alpine ecosystem. But I believe you also studied Scottish Highland cattle in the Swiss Alps for her master's and PhD. I feel a bit of a connection to those animals as someone else from the UK also living in Switzerland. Yeah, I like those ones. So today, Karen will talk to us about a fairy tale of cows and flowers. Over to you, Karen.
Once upon a time, there was a land where milk and chocolate flowed like water, and the proud farmers that raised their cows on steep mountains called it Switzerland. Far away in the north, there were some islands, windy and harsh, the British Isles. And the people of these British Isles loved to visit the Swiss mountains. Prince Charles, for example, has been there, or Sherlock Holmes or Tolkien. But a few years ago, new visitors from the Isles arrived. They came on 4 feet, and they did not consume cheese and chocolate, but grass and flowers. You know them for sure, the Scottish Highland cattle. Long hair and long horns. As grassland scientists, we found that the invasion by these cows is not a threat, but a benefit for mountain ecosystems. It requires a few words to explain why, but less than Tolkien needed to destroy his little magic ring. First, you need to know that most Swiss cows are big and heavy. And because over the last centuries, the Swiss farmers always selected the biggest cows to produce all the cheese and chocolate for the British tourists. Today, Swiss cows are twice as heavy, and they produce 3 times more milk than 100 years ago. Secondly, grasslands, on which cows naturally forage, are the home of many different plant species. Grasslands are among the most biodiverse habitats in the world. Now imagine one of these modern big cows tramples this biodiverse grassland. And like most other creatures, plants don't like being trampled. Many of them die. But some plant species are adapted to trampling pressure, and because all the other plants are dead, this plant will overgrow the entire grassland. Because farmers in Switzerland and in many other countries make their cows so productive, biodiversity is lost. In contrast, Scottish farmers did not select the bigger and more productive animals. Because the Scottish climate is too harsh and the forage is too poor for big cows, or maybe because the Scots preferred whiskey over cheese and chocolate. Either way, the Scottish Highland cattle stayed small, and they hurt the plants less. More different plant species can survive, and more insects and other animals find a home in the grasslands of the Highland cows from the windy north. And so, insects, flowers, and Scottish cows can live happily ever after.
Thank you, Karen. Thank you. I feel accused of eating a lot of chocolate, but it's
And cheese.
It's absolutely true. It's absolutely true. Okay, shall we start with Joe?
What made you so interested in studying cows?
I really love them. I think they are so calm and they are so satisfied. I always have the impression they are so satisfied with what they have, and they are great animals. They can make high quality products like meat and milk out of nothing, of a raw material which we cannot use, grass. And I think it's great that they can.
Thank you.
Tina?
I just wanted to ask the same thing, but I really thought, how do you find yes,
I can give the
cow
same-
as a
the same
topic?
answer the second time.
No. I'm just interested. I mean, you mentioned that it is also a question of biodiversity.
Yes, it is.
So, do you collaborate with others?
Yeah, of course. Because for farmers, it is not a question of, or is it a biodiversity question, or is it an economic question or productive question? A farmer has to take a single decision at the end of the day. And so it is most important that we collaborate and answer all these questions, because the Highland cattle are very nice for biodiversity, but they are very lowly productive. And so we also have to answer other questions about how can farmers live on that. Yeah, of course, we have to collaborate, yeah.
Interesting. Nick?
Kudos on that glove. That was awesome when you switch your hand like that. And also thank you for waxing lyrical about cows. I've never heard anybody talk about cows like that. That was amazing. That just changed my perspective completely on what a cow is. The question I had is, you spoke about the loss, the imbalances in biodiversity because of growing cattle. What solutions or what approaches what could we do to reverse that or to reestablish that kind of equilibrium?
Yeah, it would be nice if we all have only those Highland cattle, or rather lowly productive cattle, because there are other impacts of cattle to the biodiversity. It's not only the trampling effect that decreases the biodiversity, but of course, if we did so, we could eat about 1/3 of the meat and milk we consume today. So one way would be to reduce our consumption of animal products, and I think it's most important that we save some places, for example in the Alps, where we still have the large biodiversity, because in the lowlands most of the biodiversity is already gone. And so yeah, it's, I think a policy already does it in Switzerland to honor this and to pay farmers for to use these pastures, these grasslands with low intensity, and this helps to save the biodiversity. Okay. And that's the reason why farmers in Switzerland can live on Highland cattle, and they cannot in other countries. Yeah.
Excellent. Thanks again, Karen. So that's it for the talks. And at this stage, I want to thank you all very, very much for a really fantastic series of talks. We've all had our minds expertly expanded, I think, and we've seen some really interesting science this afternoon. So if you don't mind, can you join me in a huge round of applause for all of our finalists? Thank you. So, all right. So now it's time for the panel to discuss amongst themselves to decide who our winner is going to be. So I'll try to explain what's going to happen now. I think either the judges quietly sit there and discuss amongst themselves, or you can leave the room for a little bit if you prefer. And just to mention that the winner of today's final is going to go forward to FameLab International at the end of November to represent Switzerland on the world stage. So best of luck to everybody, and we'll see the judges in a little bit. Thank you, judges. We have to keep busy in the meantime, everybody. So while the jury debates, it's also time for those of you at home to cast your vote for who our public champion is going to be today. So we have the slide up here. There's a QR code and a link to menti.com. So there's a specific voting code for this FameLab final. So you can go there and register your vote for your favorite that you've seen today. And the voting will stay open, I think, for the next 15 minutes or so while the judges debate. So you've got to be snappy, get your votes in there quickly. Now, I believe while the votes roll in from our audience, we're actually going to take some questions from our online viewers on YouTube for the contestants. So if the contestants don't mind hanging around in the meantime, I believe I'm going to be sent some questions for you from YouTube via WhatsApp. Very high tech. Let's see if it works. But we can also take questions from the room. We can do it the Stone Age way as well, if that doesn't work. So let's see. Let's see if we have any that have come through yet. None just yet. People are maybe shy. Do we have eyes on the YouTube feed for questions? I think one of the organizers was supposed to have an eye on YouTube, but maybe not. Yes, please.
I have a question to Carolyn. Carolyn, how do you?
I think your mics are still live, so if you want to just sit and answer, then it should be fine.
All right. So Carolyn, you're standing in the lake. There's a curious fish catching with its genes. How do you even catch it? And how do you catch the fish that's shy and stays 5 meters away from you? How does it work?
I could do this myself, but I'm not a super professional fish catcher. I need to admit. So when I'm in Lake Tanganyika, at Lake Tanganyika, I work together with some local divers who are really good in particularly catching all the fish I tell them. So just show them a picture of them, and like, I need this fish, and then they bring me like 10 fish of them. So it's unfortunately, I'm not the one doing this, but I just test them. Works quite well.
Okay, but it's fascinating to know that it's actually like people, it's cool. That's really cool.
Do you study one of these 250 types, or because they're each species, right? There's many cichlids.
Yeah, so this is like this huge adaptive radiation, what you call it. The cool thing about it is that all these species are only in that lake, and they're all related to each other, so they all have this common ancestor. And what we actually do, we study speciation, and we want to know why is there such a huge biodiversity and stuff. So we try to get the whole picture of this entire radiation, but I didn't have time to study them all, 250 species, a lot. So from the whole radiation, I did like 57 species that are really nicely distributed across the whole phylogeny, across the whole tree, so yeah.
Apparently, everyone is so impressed on YouTube that they don't have any questions. So, so we can just sit in silence for 10 minutes and think about all the talks we've just seen. Question? Great. For me? Oh, no. That's outside the rule book. Yeah. I did this many years ago. I was the Swiss champion, but I couldn't remember my talk if you told me to do it now.
I was asking you, what was your talk about?
My talk was about the standard model of particle physics explained using Play-Doh. Yeah, it seemed to work, so I don't know. Yeah. So I'm a particle physicist, as you might have guessed. And the idea was to try and visualize these tiny particles in cute clay form to try and convey what it is that goes on at the microscale. So, yeah, 2013. I think it's still up on YouTube. But you've all been much more impressive than me. I'm extremely impressed at the level today. I think you've all done a fantastic job. Yeah, very, very interesting and diverse range of topics. And poor Hugo on his own is the only guy as well. So it's great to see so many women representing science. You know, it's really excellent to see that. Girl power. Question, Caroline, yeah?
I have a question back to Anna. So I saw you see me final talk, and you're like talking about all these Marvel movie-like scenes. Can you sleep in the evening thinking that this is all happening in your own body?
I actually sleep very well at night, knowing that there are all these superheroes protecting me all the time from all the dangerous
I'm off screen. It's just me looking awkward and standing around anyway, so I don't think they're missing much on YouTube. Any other questions from amongst the room? Yeah?
I'd actually like to know last year's winner of Swiss FameLab, how his last year was, and how the finals were, and what did you experience since you participated in FameLab?
The Swiss finals you mean, right?
Mm-hmm.
How was the experience? I think the setup was quite similar, I would say, last year's. Like it's kind of a hybrid session. I mean, what was interesting was, I think I was lucky in one way because last year I kind of experienced all the three possible types of audience you can experience. So I think for the Lausanne semi-finals, it was just few days before the lockdown happened. So that was the live audience, only with Lausanne. It didn't happen with Zurich and Basel. So I was lucky there. Then the Swiss finals in Bern was with a hybrid type, like you have some live audience and some online, and then you have the international finals which was completely online. So the thing, I think what was the challenge was to adapt to the three different types of audiences because the requirements were different and what you think you want to say and then what finally appears to the when I finally saw my video, I was like, what were they doing on there? So things like that happened. So I think adjusting to all these things were challenging. But on the whole, I think what was really the highlight was it was really great fun. I mean, if I just now look back at my journey and like you meet so many different people, you just hear this. It was nice to be on this audience side, you know, to chill out and actually hear to your talks. So it's like there's a lot to learn, I think, from here. And most importantly, like one thing I feel and I always say about this is like I find always FameLab to just not end with the competition itself. I think it kind of gives you a family which we kind of take along with you. We are still in contact with many of the participants of previous to previous year. We kind of sometimes manage to do some activities together. So I think it's pretty. I think it's a pretty big network. So there is a lot to experience over here. So like my message to you all would be just stay connected and like keep like track of what's happening in and around with all the participants. There's a lot to explore in here.
Good advice, I think. It really is an international family. Here I am 8 years later back in the FameLab fold after I did it. So yeah, they never really let go of you. Rope you back in to do something or other. So yeah. And last year it was all fully virtual. So there was no even, how did it work with props and things like that? People had to do it from home, I guess. You had to give your talk from home.
Finals were complete. Both the international semi-finals as well as the international finals were online. So, and it was very tricky because along with the talks, you also have to take a lot of care for the type of microphone you're using, the type of camera you're using, the angles which you are taking along. I remember we had had to do separate sessions only to make sure that their camera angle is right, my voice is clear, the recording is fine, and all these things. The internet connection doesn't even stop for a second because you're doing something, suddenly it stops and you're and so that all these things don't happen. So I think, but it was also nice to learn, just at that moment, how you interact with it. And one really good thing I found, the downside was, yes, we couldn't get to Cheltenham, to actually see the festival. That was really downside, unfortunately. But the plus side was, I don't think the viewership of last year's FameLab reached an all-time high. I mean, as many as 75 countries.
Sure.
So many people. I think I know my people here from Switzerland, my family from India, someone from Australia. It was different time zones, everyone watching it at the same time. The comments channel, people commenting in, and everyone asking. I think that was a very unique feeling to have. To just connect live with that, and that too at a time which was it was obviously not a good time for all of us. It was a very hard time you were facing. So to have it during that time, I think it was very special.
You're like the people's champion, because so many people tuned in from all over the world when this was going on. So many congratulations, and it's amazing that you came from Switzerland. Very inspiring. Great. Any more questions? Anyone have any? Yeah.
For you, with the blue poop.
That's going to stick.
I just wanted to say that, So No, but I had a question. So you were talking about the gut biome, and obviously I'm interested in food as well. So I was just quite curious. We talk so often about, oh, eat probiotics and eat yogurt and stuff, but what foods actually make your gut better or make your like little healthy family better?
So it's really what you eat. Like if you eat things that you actually know that they're not good for you, you're also letting growth of unhealthy microbes. The thing is that with all these yogurts and all these probiotic things, they can help you if you start building them up. Like, for example, after antibiotic treatment, if you had to take it orally, everything is like everything is empty in the moment, and then there's possibilities that harmful bacteria come in it. Then they can help. But if you're eating them, you would need to eat them every day and a lot to change something. And it's easier to just change your diet or adjust your diet instead of like trying something out that actually is just a marketing thing.
You mentioned earlier that very short transit times can also be bad. So there's somehow extremes of how long it takes you to see a blue poop are bad. So what can, I suppose going to the toilet too quickly, that's on the diarrhea end of the spectrum, is it? Not to get too gross, but
yeah, but would you say like after 14 hours that you have diarrhea? You wouldn't probably. Diarrhea is like every 2 hours, I guess, something like that?
I don't know. Is it?
Yeah.
It's that quick, right? Okay.
So like if you were in between these 10 and 14 hours, you wouldn't think that anything is going wrong. But to check it, it's like this first indicator saying, hey, something is not that well, and maybe you should go and check it out. And then there's like this connection when you check your microbiota then, what kind of diseases or what of risk of diseases can be connected to that.
Very interesting. Sylvie?
I just have one connecting question to that. Are there any specific diseases connected to longer times, transit times in the gut, and specific diseases more associated with shorter gut times, or is it unspecific?
Maybe I can repeat just so people hear it online. So are there any diseases specifically related to longer or shorter transit times in particular?
Like this study is going on right now. It's a huge study in England, which is going on, where they see that these biomarkers connect. So it's the one, for example, with cardiometabolic health, that you have this one bacterium which is very high, and then the people also have a very long transit time. And very long transit time is general that if food is stuck in you for a really long time, there's a lot of bacteria working on that. So you generally have a high number of different microorganisms. And it's kind of against this concept where you say like diversity is good. Diversity is good, but not too much diversity. So it's like they're beginning to see like that there are biomarkers in the microbiota, which are also seen in people that have certain diseases. It's in the process still, so they're figuring it out. In like 2-3 years, they can tell you more.
I'll ask you again.
Yeah. But it's cool. I mean, if you could use that in the hospital or really tell your patient, hey, eat the blue muffin and tell me in 2 days when it got out, and then we can talk what we're going to do next. That's very cool instead of like doing crazy tests with MRIs or I don't know what.
Sure. Thanks. I've come up with a question or two. The weather, I read recently, I don't know if this is true or not, but you're the person to answer it for me, that weather forecasters often overestimate the probability of rain just in case it happens, because if they underestimate it and then it does rain, everyone will be very angry at them. So it's better if they say, yeah, it's definitely going to rain, and then it doesn't, because no one worries if that's the case. Is that true?
I heard this rumor as well.
Some dirty weather forecaster.
When I was defending, that was my master thesis was about weather forecasting, and when I was defending it, I think 2 persons in the audience asked me exactly the same question. And I was like, yeah, I mean, the people from Meteor's Rights, they are talking with me, but I'm not sure if they would tell me. So in the end, it's a rumor. I can't help you.
You can't confirm or deny it.
No,
it's a very political answer.
not.
Yeah.
I have a follow-up question to that.
Sure.
So I also heard this rumor that when it says that when it says 60%, like, you know, it'll say on Meteo Swiss, like, okay, you know, Monday 60% like rain.
Yeah.
Is that 60% of the city will get rain, or there's a 60% chance that it will rain?
I'm actually not 100%
Okay.
sure what they refer to, but I would assume that, I mean, I was talking about these probabilistic things, right?
Yeah.
And if you just assume you have 10 scenarios, and in 6 of 10 it's raining.
Okay.
But I'm actually, I mean, it's obviously a lot more complicated than I explained today, and also this percentage is probably connecting to something way more complicated, but maybe that's kind of a easy thing you can just have, like, in your mind.
Yeah, we can take both. Yeah, Dubravka.
So I heard this thing that you cannot actually, if you see like in 3 days it's going to rain, you cannot really trust this. Everything which is more than 2 days, it's like unreliable.
Yeah, I mean, it's getting harder and harder. I mean, if you assume that you do this whole simulation of the atmosphere, which is the most accurate point, like in 10 minutes,
Yeah.
and as longer as you go, that is kind of this rule with these 3 days where, I mean, sometimes it's like more or less the big horizon which can be given already. And I mean, they are making a lot of effort to getting better and better weather forecasts, but it's really hard. I mean, especially in summer with thunderstorms, for example, they are really hard to forecast correctly.
The maths is non-linear, isn't it? Eventually, it just starts to be probabilistic, and you don't know what's going to happen.
And there is this example with the storm in California, which wasn't forecasted because a butterfly was like flying around, and they weren't accounting for that. It's like the whole atmosphere is a very non-linear, very complicated thing, like a chaotic system. So if you just like miss one little thing, like a butterfly, everything can go wrong.
Interesting. Harini, you had a question as well?
Yeah, so I was curious. You were saying that your data points are friends, but also went on to say that they're actually weather stations in the mountains and whatever else. So how much of, I don't know if this is true, this is also a rumor, the rumor mill is...
That's how science works all of a sudden. I heard so-and-so.
Gossip was so rare during the lockdown.
No,
so I mean.
so how much of your data to like model do you actually get from like the airplanes that fly? Because I don't know if this is true, but like during COVID, because the passenger flights took a hit, I was told or read that a lot of the data was missing. And if indeed this data is missing, how much of the data that you need to predict is actually missing?
To be honest, I have no idea. I
Okay.
was working with the stations, and it was really kind of this project which was just like connected to using a lot of statistics on this data, and so I can't tell. Anything I would say would be a pure guess.
Okay.
So I will leave it.
Okay, cool.
Unlike the rest of the weather.
Sorry?
Which isn't a pure guess.
No.
That's what we learned today. Yes?
Is the climate change making the simulations harder?
I actually also can't answer to this question without guessing because...
That's a good question.
Yeah, I mean, it's a really, really good question, and I wouldn't be like so rude. No, just like, I mean, there are a lot of people who studied this, and I'm a statistician by training, so it's like too hard to tell for me. But it's a really, really interesting question, of course.
Hugo, I had a question on spaghetti. I know that you do AI in your job, but now you're a spaghetti guy to me. So do you have to push it at the ends of the spaghetti? Because I could imagine taking a stick of spaghetti and I can quite easily break it into two pieces in the middle. So it depends where you apply the force. Is that what it is?
Yeah, like a spaghetti if you want to try by yourself.
Oh, so you brought many of them. So should we? It was a blatant excuse to try this. I'm gluten intolerant, so I don't get to handle real spaghetti very often. Any chance?
Just pass the box around.
Yeah, I mean, we have to clean up afterwards, but...
Who will clean up?
Okay, so we can't, sorry, here you go.
Exotic food together.
Exotic food. I shall try one on camera. A live demo of
I
physics.
just bend it.
Yeah, 3. Can we get?
Oh.
So is the rule it's at least 3, because that was 4.
At least 3.
At least 3. Should we make a histogram of the outcomes? Yeah, every time. Whereas, obviously, if you do that.
Yeah, if you do that, it's
Less pieces you get from
Yeah, sure. It needs, there's some approximation involved in the theory. Yeah.
I have a short piece here. Oh, so there's some length, there's some minimum length as well.
Yeah.
This is how science is done. Nobody's bothered to collect the data though.
Try 100 times.
Exactly. Exactly. Have our judges come back? Yes, indeed. Welcome back, judges. Hello. Okay, so should we communicate about the audience voting as well? Have you sent me? You've sent me the outcome. Very good. So I think I might do the audience vote first. Is that okay? We can do it that way. So our panel have returned, and now it's time to find out the results. So welcome back to the judges. But before we get into that, I would like to turn to the audience vote. So we also have the results from our audience online. If my phone works, it may not have, so I might have to consult. Give me a sec. I didn't get any message. Maybe the signal isn't working.
Okay, there's one. Okay, I'm clear.
Yeah.
Hang on a second. Sorry? There you are. Okay. Actually, yes, you have to know.
It will show up on the screen. Sorry, I wasn't aware that it would be on the screen, but it's Prina Yadav. Prina, congratulations. Is she with us? Prina, come on up and accept your congratulations. Sorry for the technical glitch. I didn't know it was going to show up on the screen, actually. But well done. There you go, our audience winner. Yeah, you are well appreciated online. There we go.
Thanks for voting.
Yeah, thanks to the online audience for your participation, and you also get a little prize as well, Prina. Well done.
Thank you.
I think it was those very nice bat cells, huge cells that you made. They definitely swung it, I think. Excellent job. Well done, Prina. Fantastic. Yeah. Excellent. Well done to Prina, our audience champion this year in FameLab Switzerland 2021. So now that our panel are back, we have to get down to the business of finding out who our winner of the Swiss final is this year. So as they ask in courtrooms on TV, Joe, Nick, and Tina, have you reached a decision on which you all agree?
We have, Your Honor.
Yes, we
You
have.
have indeed. So, who would like to tell me the news, Dan? Or is it going to show up on the screen? I'm not sure.
So we have decided to every one of us will say something.
Yeah, sure.
And I would start with number 3. And it's really a great pleasure to announce our number 3, which is Joanna.
Joanna, congratulations. Well done, Joanna. Fantastic job.
Thank you. Do I have to come up with
You can just stay there for now, I think. We'll do all of the stuff at the end. Thank you, Tina. On to number 2 then.
Okay, so on to number 2. Sorry,
we're going to give them out one at a time?
Yeah, yeah.
Okay.
Suspense.
Congratulations, Joanna. You stay, I think. Please stay. It's lonely up here. Nick.
So once again, it's with great pleasure to announce that the second winner of FameLab 2021 is Carolyn.
Woo!
Carolyn. Well done, Carolyn. Congrats.
Thank you.
And it is my great pleasure on behalf of all the judges to announce that the winner of FameLab Switzerland this year is Hugo for making...
The only guy.
Well done, Hugo. Congratulations. Great job. Here we have it. Well done, everyone. Many congratulations. Fabulous job, all of you. Really, really excellent. So there we have it. Our FameLab Switzerland Champion 2021 is Hugo Cui.
Yay.
Well done, Hugo. So Hugo is going to represent Switzerland in not too long. So at the end of November, we will have the FameLab International Final. So best of luck, Hugo, and good luck on behalf of all of us.
Thanks.
Understated. He's confident. So that just leaves me to say a few words of thanks. So a huge thank you to our judges for all of your wonderful input today, and you've helped make this event really special. Massive thanks online to our online audience, and congrats again to Prina, our online champion this year. And last but very much not least, congratulations and thank you to all of our participants and finalists today. I loved all of your talks. I've had a brilliant time. Sorry for harping on and on in between the talks, but really congratulations, and you've made this a really special occasion, and it's been a real delight to be part of it. So here's to FameLab, and hopefully a continuation in the years to come in Switzerland. Thank you very much, and goodbye, everyone. Cheers. Well done, all of you. Fantastic job. There we are. I think so. Yeah.